Hello, my name is Paul Wolf. I'm at Utah State University, and today I'm going to show you how to sample and collect plant material that's to be used later for DNA extraction in the lab. So this is specifically where you have a researcher who's based in the lab who's going to send somebody else out in the field to collect for them. So if you're the person in the field, you need to make sure you ask some questions to make sure you understand what needs to be done. Make sure you understand which part of the plant must be collected for DNA. Typically, this is young leaf material, but it may also be buds, flowers, or something else. You also need to understand how to do the sampling. Have you been asked to collect plants from a single population or multiple populations? How far apart should these populations be? How many individual plants per population? And then how far apart should those individual plants be? Some plants are clonal, and if you collect them too close together, you may be resampling the same plant. Okay, what equipment do you need? You need something that can be used to dry the plant material. And the best thing for this is silica gel. You can buy lab-grade silica gel, but you can also buy something like kitty litter. There are many different types of kitty litter. Most of them are inappropriate. Make sure that it is silica gel, sometimes that's labeled silica sand, and make sure there are no other ingredients such as perfumes or activated charcoal, just silica gel. You need Ziploc bags or some other plastic bag that can be sealed. You need a pencil, a permanent marker pen, and you need a sealable plastic box, and you also need a GPS device for georeferencing. In most or many cases, a mobile phone will work. And finally, do not forget your plant press to take your voucher specimens. So once in the field, once you know what needs to be collected, the first thing to do is to take your voucher specimen. I'll have some links at the bottom for those of you who want to see some examples how best to make a voucher specimen in a plant press. Make sure that all the identifying information is not just on the sample that you collect for DNA, but you also have that on the sample in the plant press so that there's no confusion later. So the key to getting a good sample in the field that's to be used for DNA extraction later is to get the plant material to dry as rapidly as possible. And to do that, you need a good ratio of drying agent to plant material, typically about a 10-to-1 ratio in terms of mass. So that's how much silica gel I use in a plastic bag. And typically for that, I'll only have about 5 to 10 square centimeters, that's about 1 to 2 square inches of leaf material. So typically one leaf that big is really all you need. And you can cut that in half. You can tear it with your hands in most cases. And you can, for some plants, especially if you've got a very robust leaf like this, put it directly into the silica gel. Often we're working with fairly delicate material that once it's dried out and it starts to get rubbing against the silica gel, the leaf starts to disintegrate, and getting the pieces can be difficult. So we can protect them by putting the leaf into some absorbent paper, like a coffee filter. And on that coffee filter, you can see I've written the identifying information, a unique combination of my name and the collection number. That should be sufficient, but just to be sure, I've also put the latitude and longitude, and the date would be a good idea as well. So then we fold that coffee filter over just to keep that leaf from coming out, and we place that inside the bag. And that's really all there is to it. Sometimes you'll get back an envelope with a few crystals of silica gel and then stuff with 20 leaves. That's not very good. You do not need more than a single leaf, a small leaf, a square inch, 5 to 10 square centimeters of most species of young leaf tissue to get the sufficient amount of DNA. So plenty of silica gel and not too much leaf. What I do then typically is from one sampling site, I will put all the bags together in one sealable plastic container, and sometimes they'll put a little bit of extra silica gel in there. This isn't the only way to collect. If you are in a moist area, if it's raining, keeping the individual samples in plastic bags is a good idea, but if you're in a desert or dry area, there are alternatives. You can have your sealable box full of silica gel, and then you could collect your samples into individual envelopes. And so the identifying information is now on the envelope. There's no silica gel in the envelope, and the leaf is protected now in there. This works, as I said, in a desert or an area where it's not raining or too humid. Okay, so the 4 main key points. 1, make sure you understand from the researcher what needs to be sampled. 2, make sure that you have a voucher specimen. 3, make sure that your sample for DNA is protected both from moisture using sufficient amounts of silica gel, and then also protected from the silica gel by having an envelope or coffee filter. And finally, make sure that all your samples are labeled clearly with your name, perhaps a unique number, the date, the species, and then the locality information. And always make sure that that information is backed up somewhere, not just on the sample, but also in a field notebook or a spreadsheet. So if you have any questions, please feel free to email me at the address below. If you have any suggestions for alternatives for collecting in the field or for improving this video, please email me. Thank you very much.